So today we've got two users joining us from Purdue. We have Craig Goergen joining us as an assistant professor of biomedical engineering at Purdue, and Craig comes from a long background with several degrees in biomedical engineering and bioengineering, as well as some time in optical imaging at Harvard as well. Craig's done a lot of really interesting work in imaging and bioengineering, biomedical engineering, image processing, image acquisition, and all kinds of really interesting deep work. He's currently on the faculty at Purdue University. Also, along with Craig, we have one of Craig's PhD candidates, Arvin Soepriatna. He's also, again, at Purdue in Craig Goergen's lab. Again, comes from a background of biomedical engineering and electrical engineering through a number of different educational pursuits. And he's working right now on developing automated image segmentation, identifying regions of interest in ultrasound. So Craig and Arvin have been working away with the Vivo 2100 system, and have been doing some really interesting work to develop images and doing gated methodologies for image acquisition. So we'll pass the presenter rights over to Craig and Arvin now at Purdue, and let them carry through with their presentation and tell us about the really interesting work that they've been doing. So over to you, Craig and Arvin.
Hey everybody, this is Craig Goergen. Thanks so much, Stephen, for the introduction. Yes, as Stephen said, I'm assistant professor of biomedical engineering here at Purdue University in West Lafayette, Indiana. My group uses small animal imaging to understand cardiovascular disease for a wide variety of applications, and we have access to a VisualSonics Vivo 2100 system that we'll be describing in a little more detail later on. Specifically today, I'm going to have Arvin, who's a second-year graduate student, be helping describe some of our recent work to look at cardiac and respiratory-gated volumetric murine ultrasound imaging, or 4D imaging, to look at both pulsation and motion of the left ventricle in the heart, as well as the abdominal aorta, both in healthy and disease states, specifically for abdominal aortic aneurysms. So just to provide a little bit of background, I know many of you are in the cardiovascular area, but cardiac and vascular disease is the leading cause of death worldwide. You can see a graph of over 37% of all deaths are related to myocardial infarction, to stroke, atherosclerosis, peripheral artery disease, a whole gamut of cardiovascular diseases beyond just atherosclerosis and coronary disease itself. We like using imaging in the laboratory, specifically for small animals, because it's very translational. It can give us information on geometry, how things appear within the body, location of disease, as well as dynamics, motion. And that's one of the great things about ultrasound. It's the high temporal resolution and fast imaging that we can do with this technique. So, right, our lab uses small animal ultrasound to look at human models, models of human disease. And the idea here is that not what happens in mice and rats is the same as to what's happening in humans, but the underlying gene contributions and pathology, the ability to identify drug targets or develop therapies are all helping us translate that knowledge into what we can use in humans to treat disease and develop and evaluate therapeutic candidates. So with that, we're going to talk about three things for the webinar today. First, methods focused on four-dimensional or gated three-dimensional ultrasound imaging with our VisualSonics Vivo 2100 system. The first application will then be for cardiac imaging, looking at mouse hearts, specifically the left ventricle. And then lastly, I'll talk a little bit more about abdominal aortic aneurysm imaging in relation to pulsation of the aorta and dilation of the vessel through this pathological disease process. But with that, I'll turn this over now to Arvin Soepriatna, the graduate student in my group, who will talk about the first two bullet points in this outline and take the webinar from here. Thanks, Arvin.
Thank you, Dr. Goergen, for the introduction. As Dr. Goergen just mentioned, I will be discussing the specifics behind our cardiac and respiratory-gated volumetric ultrasound technique, or as we would like to call it, the 4D imaging technique. We call it the 4D imaging technique because both cardiac and respiratory gating can be implemented to acquire 3D volumetric data set over time. Before delving deep into the details of the protocol, I would actually like to go over the different VisualSonics tool that we use to accomplish this goal. For image acquisition, our lab uses the VIVO 2100 small animal ultrasound system, which is shown on the right, with a 40 megahertz transducer. We decided to use a 40 megahertz transducer because it gives us the necessary imaging depth to capture the entirety of the mouse heart and its vasculature, specifically the thoracic aorta, abdominal aorta. However, I just wanted to emphasize that this procedure will work with any of the VisualSonics transducer. We collected the videos using the EKV module installed on our ultrasound system instead of the more commonly used brightness mode or B mode. The advantages of the EKV mode over the B mode approach will be discussed in more details in the next slide. We also use the 3D motor to translate the transducer along the region of interest, and we use the Vivo client host tool together with a MATLAB script to control the movement of the 3D motor. I want to mention that we can manually adjust the position of the 3D motor using the Vivo 21 system by itself, and that the Vivo client host tool is an optional part of the procedure that we use for simplicity to automate the entire image acquisition process. Lastly, I want to briefly go over the subjects that we use for our cardiac and abdominal aortic aneurysm imaging. For cardiac imaging, we used 5 male wild-type C57 black 6 mice. For AAA imaging, we used 5 male transgenic mice that are apolipoprotein E deficient and are implanted subcutaneously with angiotensin II pumps to induce dissecting aortic aneurysms. So I want to briefly discuss the advantages of the EKV module that we use over the standard V-mode approach. EKV stands for ECG-gated kilohertz visualization, and it is a cardiac and respiratory gating technique used to acquire videos or CNA loops with very high temporal resolution. Compared with standard B mode, which acquires images at approximately 78 frames per second, EKV can acquire images at a much faster rate, 1,000 frames per second. Since an average heart rate of a mouse ranges from 400 to 600 beats per minute, a standard B mode can only capture approximately 8 images of the heart along a single heartbeat, whereas the EKV module can actually capture roughly 100 images. You can actually see this by comparing the 2 CNA loops of the left ventricle acquired using B mode and EKV. In these CNA loops, the left ventricle is oriented in such a way that the aortic outflow tract is positioned to the left of the image. Can you see to the left of the image, and the apex is located to the right of the image. You can see by simply comparing the 2 videos that the EKV mode better captures the dynamic of the LV because it captures more images along a single cardiac cycle. You can also roughly visualize the general flow of the blood in the left ventricle using the EKV mode, which you cannot see in the B mode. Moving on, we're going to discuss more about the experimental setup that we use in our lab to acquire the 4-dimensional data sets. We first anesthetize the mouse using the isoflurane system shown on the left here. Once the mouse is anesthetized, we place the mouse in a supine position on the heated stage and then place its nose to a nose cone connected to an anesthesia tube. We also secured its paws on gold-plated ECG leads to obtain the ECG signals. We then remove the hair from the region of interest using a topical depilatory cream and applied phthalic ointment to prevent the eyes from drying out. We also monitored the temperature of the mouse using a rectal probe. Once the mouse has been prepped for imaging, we applied warm ultrasound gel to act as a bridge between the transducer and the mouse. You can see from the picture that the 40 megahertz transducer is connected to a 3D motor that can be controlled remotely using the Vivo client host tool. This slide gives us a closer look at the experimental setup that we used for our cardiac and AAA imaging. For cardiac imaging, we typically tilt the stage slightly to image the heart from the left side of the mouse so that we can avoid artifacts from the sternum. We then acquired serial short axis videos looking at cross-sectional areas of the left ventricle acquired from the apex to the base of the heart. For AAA imaging, we acquired serial long axis videos of the abdominal aorta instead of the short axis orientation that we used for the left ventricle. We primarily did this to minimize image acquisition time, as the aorta is much longer than it is wide. For both cardiac and AAA imaging, we translate the 3D motor with step sizes of approximately 100 microns. Since a single EKV video takes approximately 30 seconds to acquire, we need to be wary of the step sizes that we choose, since this is the biggest factor in determining the total image acquisition time. The smaller the step size, obviously, the longer the procedure will take to acquire a full 4D data set. This is why we need to closely monitor the mouse's vital and adjust the isoflurane levels accordingly. We typically start with around 2% isoflurane at the beginning of the image acquisition, and we constantly adjust this value throughout the procedure to maintain a heart rate of approximately 500 beats per minute and a respiration rate of 80 breaths per minute. We also try our best to keep the mouse's temperature around 35 degrees Celsius. Now I'm going to talk more about how we use the MATLAB script and the client host tool to automate the 4D acquisition technique. The video on the right shows an example of the automated 4D acquisition process. If you look very closely at the video, you can actually see that the motor will shift positions and acquire a CNA loop file. For visual purposes, in this video, I have selected a very large step size and acquired a simple B-mode image as opposed to the EKV CNA loops that we use in our protocol. In our MATLAB script, we actually allowed it so that it can manually select a beginning and an ending position that we want the motor to traverse to, and then we define a step size for the motor to move. And when we wrote the MATLAB code so that it automatically collects the EKV CNA loops at each spatial positions, translate the motor, and then label the file appropriately. We primarily just use this so that we have more time to monitor the mouse's vital more closely and make sure that the mouse stays alive at the end of imaging, as this is usually a very pretty long image acquisition process, which ranges from 45 minutes to 60 minutes, depending on your step size and the number of the dimension that you want it to traverse through. Before explaining how a 4D dataset is reconstructed, I'm going to briefly discuss how cardiac and respiratory gating can be used to collect 3D volumetric datasets. Cardiac gating can be used to acquire 2D images at specific time points along the cardiac cycle. For example, the peak of the QRS complex shown here. Once a 2D static image at a particular time is collected, the 3D motor can then be used to move the transducer to its next spatial position to obtain another 2D static image acquired at the exact same time point as the original image. Applying this in succession gives you a stack of 2D images that are temporally synced. These stacks of 2D images can then be merged together to create a three-dimensional volumetric representation of the heart or abdominal aorta, depending on where you're imaging, at a specific time point along the cardiac cycle. Respiratory gating is applied simply to minimize motion artifacts caused by the mouse's breathing. The proposed 4D ultrasound technique is actually just an extension of the previously discussed 3D imaging technique. Instead of acquiring static 2D images at a specific time point along the ECG cycle, we actually acquire 2D CNA loops or videos at all time points along a single cardiac cycle. Using EKV mode, we are basically acquiring these images at a rate of 1,000 frames per second, meaning that we can obtain 2D images at approximately 100 different time points along a single cardiac cycle. We can then translate the 3D motor to the next position and acquire similar 2D CNA loops at each spatial position. Applying this in succession will give you a series of 2D CNA loops corresponding to different spatial positions. Now that we have both temporal and spatial information, we can temporally sync the data across the different spatial positions to acquire 3D images at different time points through the cardiac cycle. Then you can concatenate all of these 3D files to create a 4D dataset or 3D volumetric dataset over time. After we acquired the images, we actually did some basic image processing Since the EKV module acquires images at 1,000 frames per second, and since the number of frames per EKV loop is dependent on the heart rate of the mouse, it is very important to maintain a constant heart rate throughout the image acquisition process by adjusting the isoflurane levels. In cases where there's a mismatch in the number of frames per EKV loop, we remove extra images at the end of the EKV loop, which always corresponds to the diastolic time point, so that all of the EKV loops have the same number of frames. We don't see this as that much of a problem because based on our data analysis, we typically only remove around 2 to 3 frames if you can properly maintain a constant heart rate. We then applied a Gaussian filter both spatially and temporally to smooth the image and remove background speckle noise. The actual kernel size of the Gaussian filter is dependent on the size and dimension of the images that you take. Since the transducer that we work with has its own distinct axial, lateral, and elevational resolution, the images that we acquire actually contains anisotropic voxels. So what we did is we upsampled the data using interpolation techniques and then resampled the data so that we obtain isotropic voxels. For our cardiac data set, we resampled the data to 60 microns isotropic voxels, while for the AAA data, we resampled the data to 40 microns isotropic voxels. We decided to use a smaller isotropic voxel size for the AAA data because we want to better capture the smaller and stiff motion observed in the aneurysmal portion of the abdominal aorta. As mentioned previously, there is a trade-off between the step size and acquisition time. The smaller the step size, the longer the acquisition time, simply because you need to collect more serial images to acquire data from a specific length with smaller step size. However, with smaller step sizes, less extrapolation is needed to merge the images between adjacent slices. So if you want to get the image quality that you want and you want to acquire the image within a specific period of time, you might want to consider what would be your best optimal step size that you want to use. Now that we have established the procedure used to acquire the 4D data sets, we will continue by discussing how this 4D ultrasound imaging technique can be used in cardiac imaging. Conventional echocardiographic techniques commonly utilize motion, or M mode, and standard brightness, or B mode, to assess cardiac health. M mode, shown in the top figure here, is commonly used to track the motion of both the endocardial and epicardial wall of the left ventricle along a single line. Linear diameter measurements between endocardial walls can then be taken during systole and diastole to study the contractility of the left ventricle along a particular cross-section. On the other hand, B mode, shown on the bottom figure, is typically used in the long axis view to trace the endocardial wall of the left ventricle through the entire cardiac cycle, specifically during peak systole and end diastole. Those two, the two M mode and B mode approaches, from those images you can acquire one-dimensional and two-dimensional measurements, which are commonly used to estimate cardiac output, ejection fraction, stroke volume, and fractional shortening. These are primarily used to evaluate left ventricular function. A limitation of this approach, however, is that this approach is contingent on the geometric assumption that the left ventricle can be modeled as a stack of elliptical disks, an assumption that is often inaccurate, especially for diseased and remodeled hearts. For example, depending on the position at which the M mode and B mode is taken, regions of the heart that may exhibit regional deformities may be overlooked. In addition, these techniques do not provide us with full 3D dynamic information of the heart, an information that we are primarily most interested for our research because we primarily use small animal models to study human cardiovascular disease. So I'm going to be talking about the advantages of this, how the 4D cardiac imaging technique has advantages over the conventional techniques. So the beauty of our 4D imaging technique is that once we have collected a full data set, we can position a cursor at any point along the heart to study both the structure and dynamic of the heart from any plane that you want. This allows us to study both global and regional left ventricular dynamics, particularly in disease and infarcted regions of the heart. Another advantage of our 4D cardiac imaging technique is that we can visualize the full 3D structure of the heart over time to obtain ejection fraction measurements that is independent of geometrical assumption. Because of this, we believe that our 4D imaging technique is an improvement to the conventional echocardiographic technique and can be used to better assess cardiac function and health. Here I've shown the reconstructed 4D dataset in the sagittal, axial, and coronal plane. The intersection of the two green lines here represent the center of our 3 planes. As mentioned previously, we can adjust the position of the cursor at any location we want, and we can get any images from any perpendicular planes. Firstly, I want to mention, though, that there is a small block of information missing in the sagittal plane at the bottom right corner here. However, this occurred because halfway through the imaging, the transducer actually reinitialized itself, and the imaging depth actually returned to its original setting. However, we don't feel that this affected our results since we are only interested in the dynamics of the left ventricle, and the missing chunk is, as you can see, outside the structure of interest. If you look closely at the sagittal view, the 2 papillary muscles shown here can be seen at the bottom right section of the LV cross-section. Similarly, the rib cage artifacts can be seen in the coronal view as black and dark bands. What we did with the original reconstructed data set is that we rotated it so that we acquire more standard views of the heart, which is usually the long axis, short axis, the 4-chamber view. In the long axis view, the left ventricle is oriented with its apex located to the left and the aortic outflow tract located to the right here. The 2 papillary muscles, which is previously seen in the sagittal view, can now be more clearly seen in the short axis view of the heart. Although the right ventricle is quite difficult to see in the 4-chamber view due to sternum and ribcage artifacts, the dynamics of the left ventricular wall and the left atrium here can be clearly seen. What we did with our data to quantify the volumes of the left ventricle is we initially performed manual segmentation of the endocardial border of the left ventricle during diastole. Conventional echocardiographic techniques typically include the papillary muscles as part of the left ventricular lumen. However, because we are able to acquire a full 4D data set, what we did is we decided to exclude those papillary muscles from our measurement of LV volume and ejection fraction. We then applied wall tracking techniques on the initial segmentation using deformation fields to study the structure and dynamics of the left ventricle throughout the cardiac cycle. I'm going to be talking about how we use the deformation fields and how we use an FSL software toolbox to actually obtain a video of the whole heart without manually masking and segmenting each time point. To track the wall motion of the left ventricle throughout the cardiac cycle, we use FSL, which is a functional MRI software to calculate the 3-dimensional deformation fields between successive time points. Although FSL was originally developed to study brain scans from MRI, this software contains a large library of statistical and image processing toolbox that we can use for our ultrasound application. The deformation field toolbox, which is the toolbox that we use for our application, is very similar to the speckle tracking algorithm commonly used in ultrasound images. More specifically, the toolbox tracks how much a pixel within with a given intensity has moved between images taken at different time points. For example, let us use the brain scan here to shown on the slide as an example. Assume that the red boundary corresponds to the boundary of the brain taken at time t=1, and that the brain scan in the background corresponds to the image of the brain taken at a further time point t=2. To calculate the deformation field, the toolbox uses intensity matching, which uses the B-spline transformation and a cost function to track how much that particular pixel has traveled between the 2 different time points. After tracking for the displacement of each unique pixel, we can obtain an X and Y displacement field, which we actually use to, which we implemented in our original manual segmentation to deform the original boundaries throughout the whole cardiac cycle. So after we apply the deformation fields, we can acquire a mask of the left ventricular endocardial wall, as shown in yellow here. Notice how we can use the deformation fields to actually track the boundaries of the endocardial boundary. To assess LV function, we tracked how the left ventricular lumen and wall volumes change over time. The two images on the left here show a 3D rendering of the endocardial wall, which is shown in red, and the epicardial wall shown in blue during end diastole and peak systole. From these renderings, you can identify the location of the mitral here and the aortic valve. The volume plot shows that even though the volume of the LV lumen decreased significantly during systole to pump blood out of the heart, the volume of the myocardial wall remains relatively constant and stable, which is governed by the conservation of mass. To accurately assess LV function, we use our 4D imaging technique to quantify several commonly measured cardiac parameters. Even though our measured end diastolic volume and end systolic volume are slightly smaller than that recorded in literature, the calculated stroke volume of 30 microliters and ejection fraction, approximately 70%, matches that from literature. We believe that we are obtaining smaller end diastolic and end systolic volumes because we did not include the papillary muscles in our segmentation of the endocardial wall. Through our 4D imaging technique, we can also render a video of a beating mouse heart with the endocardial wall shown in red and the epicardial wall here shown in blue to look at the dynamics and the structure of the left ventricle. Recently in our lab, we have begun to look at aortic valve imaging as a future application to our 4D imaging technique. The two videos here shows EKV CNA loops of the aortic valves taken at both the short axis and the long axis view. Notice how in the short axis view you can see the three different cusps of the aortic valve: the right coronary cusp, the left coronary cusp, and the non-coronary cusp. Specifically during systole, you can see how these cusps open to allow blood to flow to the aortic orifice. Even though we have yet to acquire 4D datasets to study valvular dynamics, we hope that this could be a potential application of our 4D technique. Lastly, since the proposed 4D imaging technique avoids geometrical assumption in the assessment of LV function, potential future application of this technique may include studying different LV pathologies, such as an ischemic heart, a hypertrophied heart, or a heart with rhythm disorder and congenital defects. We believe that by circumventing the geometrical assumption and measuring actual ejection fraction, we were able to get a more accurate assessment of cardiac health. I will now bring the talk back to Dr. Goergen, who will talk more about our abdominal aortic aneurysm.
Great, thank you, Arvin. That was wonderful. So now switch gears a little bit to go back from cardiac to more vascular imaging techniques. Just for those not in the field, abdominal aortic aneurysms are defined as a pathological dilation of at least 50% over the normal diameter of the aorta. Most of these develop in the infrarenal section below the kidneys and just above the iliac bifurcation, and they're really fairly prevalent. Around 5 to 8% of the population over 65 years of age have these aneurysms. The real problem is that once an aneurysm grows, the wall stress can lead to ruptured aneurysms. The vessel actually breaks, and with that you get a large hemorrhage and a 90% mortality rate. Most of these patients don't even make it to the hospital. And unfortunately, if you know that you have a small AAA, there's really not much clinicians can do other than wait, watch, monitor the progression, and eventually prescribe surgery, either open surgery or endovascular approaches. But there's really no way currently to prevent an aneurysm from expanding, which is one of the reasons we developed animal models to study this disease. Really the most common animal model Arvin had mentioned before is this apolipoprotein hyperlipidemic mouse that when infused with angiotensin II over a 2 to 4 week period forms these suprarenal dissecting aortic aneurysms. So these are a picture of the osmotic pump that's filled with angiotensin II. We can subcutaneously implant it into the back of the animal, sew up the incision, and then these are the ApoE knockout mice on a C57 Black 6 background. And the great thing about this model is not only has our group, and the people that developed this at Kentucky, Alan Daugherty and colleagues, but probably dozens of research labs across the world have used this, and it's very repeatable not only in when it forms, but also where it forms and how the vessel forms just above the right renal artery. You can see this large aneurysm, again, just above the right renal, showing expansion off to the left-hand side. Others have used ultrasound, including VisualSonics high-frequency ultrasound system, to look at vessel expansion. And the great thing with these non-invasive techniques is you can track disease progression over time. Baby Martin McNulty and colleagues, all the way back 10 years ago now, used high-frequency ultrasound to watch this vessel expansion and show that indeed both with short-axis and long-axis views you can see expansion of the lumen and some even thickening of the vessel wall after an aneurysm had formed in this suprarenal location. Our group and others have used both ultrasound and MRI to get 3D data sets of static vessels looking at vessel expansion. And what we found that typically somewhere between days 0 and 14, there's fast expansion and the elastin sheets break such that the medial and adventitial layers separate from each other, and you get this large growth that happens somewhere within the first 2 weeks. After that, from day 14 to day 28, we see continued slow expansion as the wall continues to remodel, collagen is turned over, and elastin continues to break down. So beyond the standard B-mode imaging, others have used M-mode as well to look at wall motion. So this now gives us a sense of how the vessel is pulsating throughout the cardiac cycle. We can measure both systolic and diastolic diameters this way, but because it's only a one-dimensional measurement, we have to use geometric assumptions of what the vessel looks like in the cross-sectional view, and these linear measurements can often estimate circumferential cyclic strain around the vessel, but cannot give regional strain estimates. So when we show what it looks like within an aneurysm, above in the proximal region, more near the thoracic aorta, you see this large pulsation. This large elastic artery is expanding quite a bit between diastole and systole. Whereas within the aneurysm itself, there's much more collagen. It's stiffer, it's bigger, but that pulsation decreases quite a bit. From a 3D approach, now that we can take advantage of the 3D motor as well, we can do serial short-axis views. So this is a great way to see how the vessel is expanding. The spine is back here. This is the aorta right in the middle. The mouse's stomach is just up top here. And now we get into this region as we go up towards the head of the animal where the vessel's expanded. And you can see this large lumen that eventually leads into a dissected region where here is the true lumen, and then this other area is a closed false lumen that's filled with thrombus. So ultrasound is great for us not only to get a sense of how big the vessel is, but what's inside of this aneurysm. Where is there flow? Where is there complex recirculating areas of flow? Where is thrombus starting to form? How the medial and adventitial layers have separated from each other. As we go up again, how this evolves over time. And we see a great deal of change usually between when we first diagnose the aneurysm and then that day 28 or 4-week period where the angiotensin II runs out and the vessel starts to plateau and stop expanding as much. Another long-axis view here of the aneurysm pulsating away at one stationary location gives us an idea, again, that we see a large amount of cyclic motion, both distal and proximal to the aneurysm. But here we can see separation between the medial or middle layer and adventitial or outer layer of the vessel wall. And this region where the aneurysm is much more stationary. So with that, we can look at an EKV slices. So now this is starting to get into our 4D approach. Similar to what we were doing with the cardiac imaging, we can get serial EKV data sets at different locations. So this is right at the right renal artery, down below an aneurysm. Here we can see midway through, this is actual inclusion of the medial layer into the lumen itself. Further up here, we see an open true lumen and then a closed false lumen. This is all thrombus that has started to form. And then further up towards the head in the thoracic aorta, you see large, elastic, more normal pulsation of the vessel. With these 3D data sets, without any dynamic information, we can do simple things like looking at both the true lumen and false lumen. The true lumen here is where blood is flowing in red. The open false lumen is this yellow area where there's blood as well, and then watching that change between baseline before the aneurysm is formed, when we diagnose the aneurysm, and then 7 days after, we can see how that open false lumen changes as the vessel starts to expand further. Everything else in green here is filled with clot or thrombus, and we can watch this evolution occur with our ultrasound technique. From histology, this is what others have shown as well. There's a focal breakage point in the elastin sheets. This is a Virchow van Giessen or elastic van Giessen stain shown in the black. These lamellar units in the medial layer have a focal breakage point here and here, where there's a separation then between this medial layer, this black layer, and then the outer adventitial layer on the outside. This all seems to be leftward, and this fibrin is starting to be laid down like rings in a tree as it develops and starts to remodel. The great thing with our 4D technique is now we can watch this and visualize it. So it's not going to have the same dynamic information as the left ventricle because there's not as much motion. And you can see, though, above and below, at least in this section, the distal sections, you see regions where the vessel's expanding quite a bit. There's healthy pulsation, whereas here you see much stiffer vessels. There's not as much expansion where there's collagen being laid down, both in the long axis and short axis view. Here's a coronal view as well, and then we see the same type of bulging expansion in one direction as opposed to general expansion in multiple ways. The 4D ultrasound technique then allows us to segment and using the same deformation field technique, look at pulsation and dynamic information of the aorta. And again, we see pulsation in the proximal and distal sections that's healthy, showing a large amount of cyclic motion and healthy elastic vessel behavior, whereas the aneurysm itself is much stiffer, again, more collagen, and the dynamic information or dynamic motion is much reduced. So we do things from a biomechanics standpoint is calculating strain. So this is just simple circumferential green Lagrangian cyclic strain and maximum amounts. And what we would expect is somewhere between 15 and maybe 18 or 20% in a normal healthy vessel. And we get that in that proximal region, but as soon as we hit into the aneurysm, it becomes much stiffer, which is what we were showing in the 4D data set. Further down, we get into some of the branches. The right renal and superior mesenteric arteries come off of this location, which is why we see some more stationary or less motion than proximal regions, but again, back up into more normal strain values. Another way to look at it is this data is using instantaneous cyclic strain waveforms. And so red is corresponding here to the more proximal regions. We see a large amount of strain at these high levels. And then you go down into this yellow and green level, more mid-aneurysm, and we see much stiffer, lower strain values at these peak levels. And then finally down into the distal, the blue section here comes back up into a more normal 8, 10, 12% range. So future potential applications, we can definitely use this to quantify pulsatile aortic motion and characterize wall dynamics, including how the false lumen forms and thrombus develops within it. And with that, we'd like to do more characterization of the dynamics and strain within the vessel and how that changes over time. And with that, just want to leave the audience with a few takeaway messages, both for the cardiac and vascular imaging. The way that we've done this ultrasound volumetric imaging for cardiac and respiratory gating gives us the chance to get these 4D volumetric data sets with both high spatial and temporal resolution. We think this is a great way to capture complex wall dynamics within the heart, within the left ventricle, or within the vessels themselves to accurately measure volumetric measurements, and with that, cardiac output, ejection fraction, stroke volume, or strain values within the vessels. And with that, it really, I think one of our big pushes in the lab is to now collaborate with people more in the computational field is using these images and data as inputs for biomechanical modeling, whether it's CFD, computational fluid dynamics, or more solid mechanical analysis looking at strain values within tissue to ideally evaluate LV function and AAA dynamics in these mice over time. So with that, I'd like to thank the other members of the Purdue Cardiovascular Imaging Research Laboratory. We have several graduate and undergraduate students are listed here, as well as co-advisor, Dr. Pablo Suachos in mechanical engineering here at Purdue, as well as some collaborators and great resources at VisualSonics, FUJIFILM VisualSonics, Andrew Needles, Desmond Hirschon, and Brett Hawkins. We'd like to thank our funding for the laboratory as well, American Heart Association, National Institutes of Health, NSF, and a couple other industry partners as well. And with that, I'm happy to thank everybody for their attention and answer any questions anybody has about our technique.
Okay, great. Thanks a lot, Craig and Arvin. That's a fantastic overview of some of the work that you've been doing. Really, really amazing stuff. Very inspiring, I think, for anybody that is looking into 4D and cardiovascular imaging. What we wanted to just wrap up a little bit with at the end here, we have just developed for the Vivo 3100 imaging system our own 4D mode implementation, which kind of takes the work that you guys have done in your lab, and great work that you guys have done in your lab, along with some other inspiration that we've had over the years from other users that have done similar work with their own acquisition methodologies with gating and triggering, and have developed this now for upcoming release for the Vivo 3100 imaging platform. So our slated release for 4D is going to be in February of next year. And it's kind of a blending, again, much like Craig and Arvin have done in their lab, kind of a blending of our existing 3D mode, the ECG triggering, and also the EKV mode. So you can think of it for any current users out there who have used our system, you can think of it sort of like again, a blending of that 3D and EKV, where you have an EKV acquisition at every single step of the 3D motor. So as we're stepping through the view, here you can see an example of what the acquisition looks like. As we're stepping through the 3D acquisition, we're gating and triggering and acquiring a full cardiac cycle at each position, and then averaging those out and putting them together. So we have some, I would say at this point, 3D analysis, volume reconstruction at this point on the system is there. One thing that we really need to work on, and we're certainly going to take a lot of inspiration from the work that Craig and Arvin have shown here today. We really need to build out the analysis, the measurements and analysis of the data. Look at things like 3D strain, 4D strain, three-dimensional and four-dimensional wall thicknesses, change in volume over change in time, change in wall thickness over change in time, and four-dimensional cardiovascular functional parameters. So we begin to think of sort of, again, ways of taking the existing functional analysis methodologies that we have now, those sort of traditional 2D modified Simpson's biplane method of discs type measurements that Craig and Arvin mentioned in their presentation, and really starting to work those into 4D space now, and look at these changes in this function over the change in time with the animals. So as I mentioned, this is going to be on the 3100 imaging system, so this is just a quick screenshot of what the acquisition dialog looks like. It's going to be, again, the Vivo 3100 is a touchscreen-based system. It's very easy to set up. Just set your 3D motor position, your scan distance, your step size, and your acquisition rates and quality, and hit go. In development right now, we're looking at, you can see there's a little estimated 5 minutes, 50 seconds there. That's for very sort of higher quality, higher frame rate acquisition. We're looking at acquisition times in the range of 3 to 4 minutes, typically, as long as 6 minutes for a very, very high quality, heavily averaged acquisition, down to as low as, say, a minute and a half, minute 50, maybe just under 2 minutes for sort of a quicker acquisition with lower quality, just to sort of check your positioning. So, along with 4D mode for release in February of next year, there's a few other things coming along, and they're all kind of related to 4D, the improved 3D reconstruction that we've had to add in because of the 4D data, an ability to do dual triggering of the ECG acquisition, whereas it was only single triggering before. You could either trigger 2 systole or diastole. We've actually, along with developing 4D, we kind of discovered a way to do the 2 of them at the same time. So that's going to be, I think, something really, really interesting. And then one sort of cool feature that we're tacking on that we want everybody to play around with and give us feedback on is vivo voice. So actually being able to do basic functions on the 3100 system using voice commands. Things like scan, freeze, save clip, switch modes, various functions like that. So that's sort of the end of my part. I would open it up to any questions that the attendees and the community out there have. As I mentioned off the top, just go ahead and use the chat window within the WebEx portal to submit your questions. Right off the top, if I could ask Craig and Arvin, I'm really interested to know, as I mentioned with our 4D implementation, we've got the acquisition working really, really well right now, but we're not, I hesitate to use the word stuck on, we're kind of a little bit paused on the analysis. And I thought some of the analysis methodologies that you guys were using were really interesting. I just wondered if you could expand on that and sort of what types of measurements and calculations from the 4D data that you think are really critical for users to be able to publish with.
Sure, Stephen. So we came in with more of an MR background, which is why we ended up using that FSL software to look at deformation fields, to track the motion of both of the left ventricle and the aorta over time. And a lot of these MR tools are great because they're used to dealing with large 3D data sets. And if we could, the only real issue on our side was to make sure we got very smoothed, Gaussian filtered, isotropic voxels that would produce these nice high-resolution temporal and spatial resolution data sets. So that is by no means necessary. I think if you all up in Toronto and as part of the larger FUJIFILM company and family can come up with ways to deal with these ultrasound 3D and 4D data sets, then that would be great. We use this FSL because of the nice deformation fields, and then really the next step for us is to start looking at strain measurements, both within the wall and in the vessels themselves, looking at not just circumferential or longitudinal radial, but the whole, the multiple directions, and looking at strain from really the tensor perspective, Lagrangian strain tensors, and tracking from a 3D approach how this strain changes throughout the cycle as well as then throughout disease development. And what we're finding is with these aneurysms or with any kind of like an infarction model in these left ventricles, you're going to see regional differences, which is why this approach with both the 3D and 4D techniques is so valuable and powerful.
Oh, that's great to hear. That's really interesting. One thing I've been wondering about, I had some conversations with some other researchers at the recent AHA meeting in Orlando last week, and one of the comments was that over time we really obviously need to use landmarks. Landmarks are really critical in cardio imaging, even in 2D. In 4D, again, there's a concern over sort of using an anchor point or a landmark, especially in pathology models as the ventricle remodels and things change over time. Have you, just asked you to comment on the types of anchor points in 4D space that you're using to be able to compare your data sets over time?
Sure. So for us, really, we were finding that the pattern that we see, the speckle pattern within the wall seems to be fairly robust. And we can start using that within the papillaries, within the muscle itself, or in the aorta, the case where there's branches or in a dissecting aneurysm, a separation between the two layers, that's the markers that we tend to go off of as much as possible. We just need, the great thing about the 4D, right, is that when we tried to do this strain measurements with a 2D approach, these speckles would start moving in and out of the plane, and it really screwed up any kind of analysis that we were trying to do with more sophisticated strain measurements, either long or short axis. But with this 4D approach, those speckles now we can track regardless of where they go, whether they move in and out of the plane, because we've got the full 3D data set. So it depends on the quality of the data you're collecting. If you can get the speckle pattern within the vessel wall and track that over time, you should be able to get strain measurements from that, or you can use papillaries, mitral inflow, aortic outflow, celiac, SMA, right renal, all the different vessels as a way to figure out where you are within the vessel or in the heart.
That's good to hear. I know that was one of the concerns. Definitely was that sort of landmark. So that's very interesting. Good. Another question, just kind of actually extending on that a little bit. Craig, you mentioned that you had come earlier on. You had mentioned that you had come into this from an MR background. How do you feel the gated and triggered and averaged 4D ultrasound compares with MR data, not only just in the visualization, but in the ability to analyze and reconstruct those data sets?
Sure. I think a typical MR researcher will look at some of the artifacts that we see in ultrasound and not know what to do with it. The rib artifacts or the lung artifacts, and there is an art to it. Even the students in the group and people that use our system, there's a little bit of training. You need to know the right orientation and make sure the transducer's positioned correctly. You try to avoid all the bubbles in the gel, and there's somewhat an art to it. But once you get positioned and trained well enough, I think it rivals and if not supersedes the MR aspect. I mean, it certainly is more easy to access for a lot of groups compared to a high field, small bore animal system, MR system. It's a lot easier to operate, and the interface with the VisualSonics system, just getting things up and running is fairly simple, and acquiring these data sets, right? We were taking 30 to 45 minutes, but it sounds like the next generation with the 3100 system can be on the order of 3 to 6 minutes. That really rivals, if not beats, what we see with MR. And the data sets that, unless you really perfect a 3D volume acquisition with MRI, you're talking about 1 millimeter thick slices, and we're getting much better isotropic voxel resolution with our ultrasound. That you're going to have trade-offs with some other aspects, maybe with depth, right? You're not going to be able to go into a large animal model and get that high resolution system if you're going to need to go 5 or 6 centimeters down into large animals. But at least for the small animals, our ultrasound does great.
Great. Yeah, that's actually talking about sort of setup and acquisition times reminded me of something that you Arvin, had mentioned in your presentation about using that Vivo client host tool, and I just wanted to clarify that on the similar work could be done on the 2100, I think, without that client host tool. It's just makes it a little, makes it a lot smoother, I think, from your perspective and the work that you're doing. The implementation that we have on the 3100 system eliminates that need completely, because the 4D acquisition is a mode that's available on the system. It's much like what I showed there with the user interface on the touch screen. It's mostly set up the animal, get your position, check that you have good physio data, and then hit essentially scan on 4D mode, and you've got your data set within a few minutes. So very, as you said, Craig, compared to the way that you guys are approaching it in MR, 3 to 6 minutes tops. It's pretty quick and easy acquisition. We have another question here from a current 3100 user, just asking about a way of segmenting the endocardium and the epicardium in 3D acquisition and analysis. So there's a couple, I think there's kind of a couple questions wrapped up into one here. One is, with the 3D acquisition as it currently stands on the 3100, it's sort of more of the traditional gated, respiration-gated and triggered to either systole or diastole, independent from one another, so 2 different data sets. 3D acquisition, where you have a data set where the heart is very static in that view, so you don't have that time component in 4D. It's just purely 3D. So if you gate and trigger to systole or gate and trigger to diastole in those individual data sets, you should be able to see the endocardial and epicardial borders. You may have to move things around a little bit in 3D space and sort of change the planes of view a little bit. But the ability to kind of visualize is there, and the ability to do some volume analysis is there. But again, it's going to be static. You're not going to have that dynamic time component. The other kind of part of the question about only being able to choose endocardium or epicardium, I kind of wondered if that was about the vivo strain implementation. So the current vivo strain analysis implementation that we have, you do have the ability in 2D in either long axis or short axis to do tracings of the endocardial and epicardial border, look at longitudinal, radial, and or circumferential strain in the endocardium and the epicardium, and also, again, the radial component between the endo and epi component. That is a 2D implementation right now. We are looking down the road now that we have a really robust 4D acquisition mode, we are looking down the road to being able to implement some 4D strain. So
that'd be really helpful.
Yeah, we're really excited about that. And then I think there is just one other follow-up question to that as well about doing the tracing in 4D. So in the 4D implementation that we have now, yes, there is the ability to do volume tracings, but again, it's kind of what you saw in that video where we're doing the volume tracings in 3D and also through the time component, where you're seeing the volume move over time, and you can get volume numbers at different time points. The next step for us will be really to integrate that into a measurement tool. So I think that's probably about it, I think, for this webinar. I think we're right at 2 o'clock now at the 1 hour mark. So I do want to say again a really big thank you again to Craig and Arvin for taking the time and putting the presentation together and really showing us some really exciting work that's being done down there in Purdue. We're really looking forward to seeing this 4D acquisition mode get out into the broader research community and really seeing where our users are going to be able to take it next. So again, thanks so much, Craig and Arvin, for joining us. Great to have you.
Yeah, we appreciate it. Yeah.
And if there are any follow-up questions at any time, you can submit them to us through our website, VisualSonics.com. And webinars, there are some web forms there where you can go in and ask for support, be it application support or technical support. If you are working with 4D, if you're interested in 4D, we want to hear about it. We want to hear about the types of analyses that you would like to do. And also do keep an eye on our website, again, VisualSonics.com/webinars. This, I should mention, this is be the last webinar for 2015. So moving ahead into 2016, we'll have a whole new slate of monthly webinars to share with everybody. So thanks again all for joining us, and we'll speak to you again soon.